In my previous video, I looked at what it would take to make a self-replicating machine, a von Neumann probe. It was clear that 5 subsystems were required: materials, manufacturing, computer, energy, and information. All 5 of these subsystems exist in all living things. Let's look at them one at a time. First, the materials subsystem. The simplest form of life requires just 6 kinds of atoms: hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. That's it. This is a much simpler list than what our probe required. In fact, hydrogen and oxygen, water vapor, carbon dioxide, CO2, and nitrogen gas, N2, are abundant in the air. That said, the cell's membrane doesn't let just any molecule through it. Some molecules, such as carbon dioxide, can eke their way through the membrane. Water, however, cannot. A special channel protein system is necessary to let water through. Other atoms like phosphorus and sulfur aren't that abundant, so they need to be grabbed when available. This requires active transporter gates which draw in the necessary molecules one at a time. Each gate only lets in one type of molecule. These gates are made of protein complexes which must be manufactured by the manufacturing subsystem. Also, the transporter gates open and close based upon need. Opening and closing gates require energy, so we already need support from the energy subsystem. Once we have raw material, we need to turn it into the parts that we need, hence the manufacturing subsystem. For DNA, we need adenine, thymine, cytosine, and guanine, and the sugar phosphate backbone. RNA also requires uracil. So, how are they manufactured? According to ChatGPT, through, quote, a series of enzymic reactions. So enzymes, protein, RNA, and combinations, each perform one step in the process of building up these essential components. Breaking apart H2O, CO2, N2, etc., taking each of these atoms and creating subunits, taking the subunits and building larger subunits, finally building the final part. It is really quite like a modern assembly line. And then there's the 20 different amino acids. They also require a series of enzymic reactions. More precision proteins working hand in hand with other proteins. You may remember that to get a von Neumann probe, we needed to be able to manufacture the 3D printer that would manufacture the parts. The same here. Every protein required for every stage of the manufacture of DNA's components, and every amino acid required to build those proteins must be manufactured. And all of these proteins, all of these RNAs, are precisely built from instructions encoded in DNA. And many of these enzymic reactions require energy as an input. Bruce. Yes? These chemicals form naturally in the Miller-Urey experiment. Hold on a minute. Many of these base molecules are produced in Miller-Urey type experiments, but never with the kind of precision and volume required for life. And more importantly, no living organism uses a Miller-Urey-like technique to produce its components. Rather, all organisms need the disciplined approach of enzymic reactions using precision proteins and RNAs. So where do these proteins and RNAs come from? They come out of the 3D printer of the cell, the ribosome, with support from RNA polymerase. The ribosome is a computer that translates DNA into protein through a translation code called the genetic code. It is a very disciplined process where sets of 3 nucleotides always add a particular amino acid to the growing protein chain. The protein chain is forced by the order of its amino acids to contort itself into precise 3-dimensional parts that work hand in hand with other parts to do the manufacturing. For a long time, I had the view of an RNA polymerase chugging down the DNA, spitting out mRNAs to a ribosome which created protein. This view is incomplete. Rather, each organism has an army of RNA polymerases clamped onto different locations of the DNA, generating the mRNAs for proteins as needed. The RNA polymerases pop onto the DNA at the needed point, produce the needed mRNAs, then pop off. So the central processor of the cell is actually a fleet of parallel processors, somewhat akin to a modern multi-core CPU. And of course, RNA polymerases and ribosomes require energy to do their work. So let's have a quick look at the energy subsystem of the cell. You may remember that we considered hydraulics and pneumatics when building the von Neumann probe. We settled on electricity, but there are other options. Biology settled on ATP. ATP provides chemical energy for all of the energy-intensive functionality of the cell. In all cases, ATP is converted to ADP, releasing energy. The cell is kept well stocked with ATP so that every process that needs energy has a source at hand. The ATP can be viewed as a charged battery, where ADP can be viewed as a spent battery. There is a whole precision multi-protein manufacturing machine that converts ADP to ATP. Well, there's actually hundreds of these things in every cell. These are effectively the battery chargers of the cell. Bruce, could life have been created without ATP and ATP synthase? Well, that is $1 million question. What we do know for sure is that no life form yet has been found or created that does not use ATP generated by ATP synthase. What about viruses? They don't have ATP synthase. True, but viruses and other parasitic organisms get some or all of their essential duplication technology, including any needed ATP, from the host. The host gets ATP from ATP synthase. So all ATP in cells is generated by ATP synthase, either by the organism or by the organism's host. And that goes for the other essential subsystems. And I'll save the best for last: information. As we walked through the copious list of items that need manufacturing, each requiring precision information, we find the information subsystem to be, well, overwhelming. So here's what I've decided to do. My next video will take a look at the information in the world's simplest living organism. I bet it will amaze you. It certainly amazed me. Please like, subscribe, comment, and share this video. Thank you.